Good evening, folks. Nice to see all of you. Welcome to UCR Palm Desert. I hope you took a minute to enjoy the reception put on by our good friends at the College of Natural and Agricultural Sciences. Welcome to UCR Palm Desert Center. My name is Agam Patel. I'm the executive director here. I'm so happy to welcome each and every one of you to the new lecture series for 2025 with our friends at CNAS, College of Natural and Agricultural Sciences, the Science Lecture Series. It's a 4-part lecture series. 2 of them will be held right here, and 2 of them, I'm hoping you can make a special trip to Riverside and visit them. Each science lecture topic tackles some of the most pressing issues as we face locally, nationally, and globally. So the 2025 Science Lecture Series is sure to not disappoint you. Before we bring up the newly minted Dean for CNAS, not new anymore, he served as the interim Dean, who will intro Mary Droser momentarily. Just give me a few minutes to go over some important reminders coming up right here at UCR Palm Desert. The very next lecture in this series, March 4th, we're going to be joined by Miguel Arrieta, who is going to be talking about quantum tomography. Again, this is the second one in this very science lecture series. If you haven't already, pick up a flyer outside, but we'll also be sure to send you the reminder in our newsletters. Next week, February 11th, the second in our partnership with our friends at the ModCom, Palm Springs ModCom Committee, we are presenting architect Gary Wexler right here, who's going to be talking about the modernism movement. So that's always a popular series. If you don't already have a flyer for that, pick it up outside. Happening on the 18th is a continuation of our Wild Coachella lecture series. I know I've seen many of you attend the other ones headed by Dr. Lynn Sweet. In fact, she's going to be talking about plant communities on February 18th, so mark your calendars. And for those of you that are not aware, the Center for Conservation Biology has its offices right here at UCR Palm Desert upstairs, and it's part of our College of Natural and Agricultural Sciences. So mark your calendars for that. On the 19th, we're going to be joined by our friends, again, Tim Lyons and team. We're going to be presenting Caroline Hung, who is a PhD candidate. She's going to be talking about the environmental changes on water quality at this critical juncture for the Salton Sea. So I know I saw most of you here last week when Tim Lyons gave us an overview. This is going to be the second in that 4-part lecture series. So mark your calendars for that. Still in February, bear with me, it's a busy month. We're kicking off our Boyd Deep Canyon series. For those of you that are not aware, we've got a beautiful research facility just about 10 or 12 miles up Highway 74. I'm working with Dr. Chris Tracy to put this series on. It's been going on for 15 plus years now. We're going to be featuring Dr. William Hees, and he is going to be talking about all things desert tortoises. So pick up a flyer for the Boyd Deep Canyon. A couple more towards the end of the month. On the 25th of this month, a very special lecture, the Fay Lecture in Jury and Innovation with our friends at UCR's Jewish Studies Department. We are also going to be honoring the late, great Bob Fay, who was a community leader, a philanthropist, and an all-around champion for UCR. We're going to have a bench dedication for him. So join us for a reception at 4:30 right here, followed by a lecture at 6 o'clock. So mark your calendars for the 25th of February for that. And then on the 27th of this month, we're taking our Palm Desert Center partners to Riverside. So for those of you that are interested in becoming a partner, come see me or come see my colleague Laura, who's floating around somewhere back there. We are taking our Palm Desert Center partners for a special trip to Riverside. Check out our state-of-the-art facility with School of Medicine, followed by a reception and a lecture, and then we'll drive you back. So it's going to be a fun day. We hope to see you there. If you are a Palm Desert Center partner, I know I saw a number of you out there. Please raise your hand. I want to publicly acknowledge you. Thank you for your support. I know some of you joined just today as well. Thank you for your generosity. We couldn't do this work without you. and your continuing support means the world to us. All right, let's get started, shall we? Dean Peter Atkinson, a professor of genetics in the Department of Entomology, joined UCR in 1997. Prior to his tenure at UCR, he was a senior research scientist at the Commonwealth Scientific and Industrial Research Organization in Canberra, Australia. A first-generation college student, Atkinson earned his PhD and BS with honors from the University of Melbourne in Australia. Since joining UCR, Atkinson has served in a number of administrative roles within the college campus and the University of California system. He has served as deputy and interim director for the Institute of Integrative Genome Biology, director of the Interdepartmental Graduate Program in Cell, Molecular, and Development Biology, chair of the Institutional Biosafety Committee, divisional dean of Life Sciences, and chair of the Senate Committee on Planning and Budget. So just a few things on his spare time. Join me in welcoming Dean Atkinson.
A little more sense to you in a few minutes' time. It's a privilege to be back here after a lapse of about 10 months since our last Science Lecture Series, and it's something that we really wish to continue doing out here, investing in the area and bringing the very best of UCR faculty and their science out to you out here in the Valley. Tonight we're with Dr. Mary Droser, who is a Distinguished Professor of Geology in the Department of Earth and Planetary Sciences, and the topic of her discussion tonight is animals, sediments, slime, muck and goo, the record of Earth's early animals and their environments with implications for discovering life elsewhere. And this is astonishing work. Dr. Droser obtained her undergraduate degree at the University of Rochester and her PhD from the University of Southern California. And she's a paleoecologist, a paleobiologist who specializes in the advent or the origin of complex animal life on our planet. Now, she has done her research in her career on 4 continents, but the one that is going to come to our attention tonight is Australia. And because I was born and grew up in Australia, that means something special to me. Growing up as a child in Melbourne, Australia, which is not in South Australia, and I bring it up because Melbourne is in Victoria, and growing up there were strong feelings about other states. There weren't very few Australian states, so when we talked about football and cricket and other things, South Australia was a different state. But what we learnt as children was that Australia was a land of natural wonders. It had kangaroos, it had wombats, it had koalas. Yes, we all know that. In Victoria, South Australia, Queensland, New South Wales. But it also had some stunning natural wonders, and the Great Barrier Reef was one of those. And the other one, which we knew about even if we didn't know where the Flinders Ranges really were, that they were in South Australia as we found out, was always said, quite rightly, and you'll see some, I haven't seen them, but I know you'll see some magnificent photographs of Mary's sites out there in the Flinders Ranges, which if you know where Adelaide is and you think about driving 6 hours north into what becomes the Great Australian Outback, there is this fascinating mountain range. Now, growing up, it was a natural wonder along with the Barrier Reef. You're going to hear tonight how it's a natural wonder in a very different way than just scenic beauty. And this wonder, I would argue, and proud to know South Australians and proud to know people who work in South Australia and proud to know Americans who are putting South Australia and the Flinders Ranges on the map. Mary will probably comment that because of her work, that particular site, the Flinders Ranges, is now being proposed, work in progress, as a World Heritage Site, which once it happens means that for posterity, it's not just Australian kids learning how wonderful this natural phenomenon is in some other part of the country. It's preserved for posterity, not just because of its natural beauty, which has got nothing to do with humankind, but with the work and the discoveries that Dr. Droser has made in those ranges, which you'll hear about tonight. This is a big deal. You know about World Heritage Sites. You are witnessing, if you will, one in the genesis of becoming one, and an outstanding scientist at UC Riverside, California, United States, making such a big difference to another country and to the preservation of significant sites worldwide. So I have a bias because that's where I'm from, albeit Victoria, but seeing, yes, she's laughing, but she knows this parochialism that goes on, but actually seeing, being part of the university as a faculty member that is bringing the magnificence of this unique landscape in its natural beauty and in its scientific significance is a privilege just to be standing here to talk about it. But enough of me. I'm going to introduce Dr. Mary Droser, who's going to show you some wonderful scenery, and you have beautiful natural scenery out here. More of the same, but slightly different color and a little different, but then really about the science, which is absolutely extraordinary. So Mary, please.
Thanks, Peter, and lovely to be here. So I'm going to step away from Victoria and South Australia for a second. Every single person in this room has at one time or another wondered if we are alone. Is there life on some other planet? Whether it was, sitting around with high school friends or more recently with news of materials that make up life being found on an asteroid. We've all wondered, are we alone? With the discovery of so many exoplanets right now, the question is even more prescient as we find Earth-like planets very far away. But for now, it's an N of 1. There is only one planet that is covered with green scum, and trees and life and all that other kind of stuff that goes along with life. But at one point, of course, Earth was, like the other planets we see off in the distance, a big rock hurtling through space. Well, we're still hurtling through space, but now we have life on it, right? But the amazing thing is that we can go back in time. We have a geologic record that allows us to go back in time and look at alternative Earths, look at Earth at different times. And so we can look at the rocks and interpret them that there was about 3.5 billion years ago that this was a watery planet. Another state, Western Australia, we're very state focused today. Western Australia has the oldest evidence of life on planet Earth. It's a little squidgy thing that right over there in rocks that are about 3.5 billion years old. So we have this incredible record of life going back in time of planet Earth. I'm going to zoom way ahead from here by about almost 3 billion years. We have the origin of the Earth at about 4.5 billion years ago. We have bacteria for a long time. Then we get a little bit complex single-celled animals or single-celled organisms. But then we have the advent of animals way down there at about 550 million years ago, where we have the advent of complex animals, things with more than one cell. And that's what I'm going to talk about today, this early record of animals on planet Earth. So this is the very quick sort of summary. This is, in the US, sometimes we do dinosaur series. In Australia, they actually did a stamp series that with some of our fossils here. Here's one of our fossils right there, not this little guy here. The summary, just to give you what is it I'm talking about. So they're called the Ediacara biota. Ediacara is a local term. Actually, they were discovered in South Australia. Ediacara just is a term to characterize a group of fossils, a group of organisms. All of them are soft-bodied, so no hard parts, no bones. Think things like jellyfish and sea anemones, soft, squidgy things. They're found around the world, but at about a handful of localities, they're pretty good. This group of weird fossils, and you're going to see how weird they are, spanned about 30 million years. So it's not just a blink, it's a long period of time. And the fossils I'm going to talk about today from South Australia, they're neither the oldest fossils or the youngest fossils in this 30 million year span, but they are the most diverse and include a lot of firsts in the advent and early evolution of animal life. So just to give you an idea, when you think about, okay, this is the unfolding of animal life on Earth, these things are soft-bodied. Lots of times people are like, oh, are they microscopic? No, they can, sometimes they're centimeters, sometimes they're this big. The other thing is they're abundant. These aren't rare. You're not like out for a whole day and then, oh, I found one. When they're there, they're abundant, as you'll see. The other thing that's really interesting is that when we think about, you know, what we all learned in high school, kingdom, phylum, order, and, you know, memorizing, coming up with some acronym to memorize how animals and plants are classified, these things are so weird that we don't even classify them above the genus level. We talk about species and genera. We don't think about families or classes or orders or honestly even phyla. They're that weird. We can't, what we try to do is put them in the animal tree of life, but they're pretty weird. The other aspect of them is that most genera only have one species. So there's like the genus that is for oak trees has, you know, tons and tons of species within that one genus. Here, most genera only have one species. Here's just what they look like, not the bearded dragon here, but that's this guy there, that's my scale. They look pretty weird, right? You're like, okay, this guy here maybe looks like it has a head. This guy here, it is triradial. That means we are bilaterally symmetrical. It has like trilaterally symmetrical. Nothing living today is triradial, right? That is an extinct body plan. So when you look around, you know, that looks kind of like a leaf, except that it's not. It is an animal. And so this is just to give you an idea of sort of what we are dealing with and thinking about. Now, as Peter promised, I am going to be talking about an area that's of South Australia in the Flinders Ranges area. These are the beautiful, spectacular Flinders Ranges in the distance. And this land lies within the Adnyamathanha traditional lands, who are the rock people in South Australia. And this is South Australia. Victoria's, you know, over there a little. So, most paleontologists, when they go out in the field, I'm a paleontologist, you have a hammer, you go out, you hit a rock, you find a fossil, you take it back to the lab, you take it back to the museum, and you look at it. Right? That's how standard paleontology is done. That's not what we do here, and there's a reason for that. If you picture sort of this seafloor up here, or any seafloor, the Gulf of California, the beach off our coast, and there's lots of things living on the seafloor. What happened, I should say, is that every now and then a storm would come and just smother the whole community. And it would preserve, basically cast, it would make a cast of that community. And this was an area, sort of like more the Gulf of California, where there weren't that many storms. So a storm would come, deposit sand, and then you'd get a whole community building up again. So the storm comes, here's your organism sitting on the seafloor, it's casting the top of the organism. These are the storm beds, or each one of these And so the fossils are underneath these sandstone beds. So you have fossils underneath here. So to get at the fossils, you have to flip the beds over. So paleontologists working here before we started would sit there with hammers, take a piece off, and take it back to the lab. But we had a different idea. I was working with a guy, Jim Gehling, from the South Australia Museum at the time. Our Nilpena station was about 1,000 square kilometers, so sheep station. This is the pastoralist, or here we would say the rancher. Right there, a guy named Ross Farger. And Jim and I went up to Ross and we said, we kind of have this idea. All these fossils are underneath these beds here. There's soil on top of it, but we think we can remove that soil. So maybe we remove that soil, and maybe we can excavate these as whole beds, bits and pieces, excavate them and put them back together so that we have snapshots of the seafloor. So he said yes, which was great. And so we started doing that. Here's some of early excavation of beds a long time ago now. So this is a hugely laborious process. Basically, we're getting dirt off the top of beds, we're clearing off the surface, we take Sharpies and whatnot and mark the bed so we know how they're all together. We don't ever break pieces. The rocks were like this, they got tilted up like this, so they're broken. And so sometimes they're the size of a pinky nail, sometimes they're the size of your dining room table. But then we take them, we put the plastic down, we flip them over, and we get basically a snapshot of the Ediacaran seafloor. This takes a lot of effort. There have been a huge number of volunteers and students involved with this work for a very long time. So, to give you an idea of how long, I first went to South Australia about 25 years ago. I had an NSF grant for 2 years in South Australia, 1 year going to Namibia. This was, my husband works in the Himalayas, so the kids always came with me. This is the second year. These are my two kids. This is what they look like now. And I'm still working in South Australia. So I went for 2 years. I ended up staying 25. A lot of work to do, never left, and now, you know, it's my home away from home. We didn't know when we first started working there, A, that we could excavate, or B, that it would just be such an extraordinary fossil site. So amazingly lucky. So this is a hillside shot of our excavation. We've excavated about 40 beds. When I started this, I sort of had in the back of my head, I wonder if I would need fins in the Ediacaran. So when you go snorkeling or diving, you have fins and you like paddle over here and paddle over there. So in my head, as one does when you're in the field, you imagine yourself 550 million years ago. I went into it, that whole 2-year thing, thinking, we'll excavate some beds. They're all going to be the same. This is the Ediacaran, it's first animals. Come on, how complex can it be? 25 years later, we're still excavating very, very different beds. So as we're sort of trying to piece together, what did Earth's earliest animal ecosystem, these are the first animals on planet Earth. It is marine, by the way, it's the ocean, I should say. It was very complex pretty quickly. So in case some of you have had very long days and need to take, you know, we'll have a little drop off. I'm giving you the punchline right now to make sure you get it. So Nilpena, so it's Nilpena Station was the name of the locality. It is now Nilpena Ediacaran National Park, purchased by the government. Oldest record of active mobility, oldest bilaterian, I'll talk about that in a minute. Oldest evidence of scavenging, oldest evidence of life in the water column, oldest evidence of sexual reproduction, oldest ecdysozoan, I'll tell you what those are in a minute. Complex ecosystems and ecological interactions. The take home is not only that this is obviously an extraordinary fossil site, but also that the dawn of animal life and early animal evolution began this early, began with these weird, strange looking things in what's called the Ediacaran period. So some of you may have heard of the Cambrian explosion. We are before that. So really the opening of complex life on Earth. So I showed you this before, and you look at it and you're like, well, how do you deal with these? What do you do? So we get on our hands and knees and we map everything on there. These are Post-its, those little Post-its about the size of a finger. Everything gets mapped on the beds. So by doing this, it forces us to look at everything and understand what all is there. This is how we make our discoveries, essentially. So this is something that was described before by somebody working at a different site in the 50s who called it Form A, didn't give it a species or genus name, and he interpreted it as fecal pellets. So there's a couple reasons why that's wrong. Branching fecal pellets, just try and get that one around your head. Also, strings of fecal pellets all lined up. You're talking about animals all lining up and, at the same time. So right away we knew, like, yeah, no, not fecal pellets. And we had the advantage because we had these huge bedding planes, so we weren't just looking at one little thing, we're looking at a whole bedding plane full of these. We spent a lot of time on this. We named it Phoenicia dorthea. Phoenicia is Latin for rope because our nickname for it was ropey. It looks like a rope. We interpreted it as the body fossil of a tubular organism. And one of the really interesting things that we discovered about this fossil was about the juvenile attachments. So these are these tubes. They're sitting up, let me just go back, standing up in the water column, and they have these attachments on the base. And most of the time you see the tubes, but sometimes you see the attachments. So what we discovered about these attachments was they were densely packed, as you can see, and they occur in size cohorts, as you can see here. So that means, if you think about it, all the same size, all the same generation are all hanging out together, and they're really densely packed. This kind of growth or reproduction is called synchronous aggregate growth. Synchronous, they're all the same generation. Aggregate, they're sitting like this. Growth, they're all growing together. This is really common. You can go to our tide pools in California and see it with mussels and barnacles and so on. It also has this branching, which we interpreted as budding. So we interpreted this particular organism sort of being a cnidarian grade, like a coral type, but it's soft, remember. So we published this in Science, and we suggested that it was evidence of sexual reproduction. It was, at the time, and sort of still is, the oldest evidence of sexual reproduction. But think in terms of sort of corals on the Great Barrier Reef shedding egg and sperm into the water column because of triggers with the environment and so on. So, all the sexual reproduction, you can imagine, it went from the New York Times all the way down to the Daily Mail and got raunchier and raunchier and raunchier. I had the Dorothea is my mom. We named it after my mom because she spent years taking care of my kids when I take them there. So that became a whole thing. And in fact, she was interviewed for the London Times, and it was like, she's standing in Ontario Airport having just arrived. It's like a 45-minute interview. The only thing they put in there was when they said, well, do you believe in sexual reproduction? Weird question. And she was like, well, of course I do. I have 11 grandchildren. So, single quote. You can imagine this was, well, I was glad I had tenure when it came out. It wouldn't have mattered, I know, but still. So we're on our hands and knees, and we're looking at things, and we see rocks like this. And if you're looking at that going, oh, I don't know what I'm supposed to see, fair enough, we didn't either. What you see here is lots of what we would call textured surfaces. This is the bedding plane. This is a casting of the Ediacaran seafloor. And if you look sort of at these right here in particular, they look sort of fluidized. They look a little like fluid in sand. So remember, we're 550 million years ago. So when you go to the beach today, You have lots of things. This is my most favorite invertebrate. This is a sea cucumber, Great Barrier Reef, Queensland, another state. This thing is the size of a Wonder Bread family size, huge, right? And it's pooping. The poop is cleaner than what goes in. It's just clean sand because these guys just eat every single bit of organic that's on the seafloor. They just, nothing. And when you go to the beach today, you don't see lots of green organics. Everything is being eaten, the sediments are being burrowed, and so on. When you go back 550 million years, it looked like this. In the absence of things like sea cucumbers, in the absence of things burrowing and mixing the sediment, the seafloor was covered with pond scum. It's my title, ooh, goo, muck. You know, you would not have wanted to put your foot into it. Disgusting, right? Would have looked something like this. So that's your seafloor. These animals are all living on something like that. And so these rocks are casting that microbial surface. And it turns out that this organic mat was absolutely integral to the ecology and life of these early animals. And it was food, and some of them were just stuck in it, and was really a big part of the biomass. So lots of times you'll see it, well, not lots of times. If you ever see a diorama of the ediacaran, it usually has a green seafloor. That's from the microbial mat. And this is a very old diorama of the ediacaran, ediacrobiota. I call it the Noah's Ark version. It's 2 of everything. 3's too many, 1's too few. Totally not accurate. But I understand why they did it this way. All right, one of the things that mats are great for. One of the great things about having microbial mats on the seafloor and the ediacaran is evidence of mobility, evidence of movement. So we all got here, and that was great, and we moved around to have our buffet out there. Everybody likes to go to a restaurant and be served. But if that's the only way we could ever eat, we would all starve, right? The ability to actively move, to leave your environment and go someplace where it's better, to go and find food someplace, is a major, major deal in the evolution of animals. It's a big novelty, and very important to us. So the oldest evidence of mobility occurs in this ediacrobiota. That's a former graduate student, Scott Evans, who's now a faculty member at Florida State. This here is a fossil called Dickinsonia. This specimen is about this big, but they can be the size of a pinky nail. They can be the size of your bath mat. Remember, this is soft-bodied and it's like squeeging all over the seafloor. So this guy moved. We know because we have evidence of it moving. We have evidence of what we call footprints. It's a terrible name. They didn't have feet, but it's just, and it's not our name, but it's a name that's used. So here's our organism. Here it is sitting in the microbial mat. That's this green here. It ate through its underside, right? So it expanded a little bit, and we can tell this from aspects of the fossil record, and then absorbed food from its underside. So it would hang out there and eat, and then it would get up, probably just not like get up from a chair, but actually lift up from the seafloor and go to another spot. And what it would do is leave this divot in the microbial mat. So a storm comes, it casts the top of the Dickinsonia, and then it fills in this divot here. This is crazy, right? That we can pick out this kind of detail. 550 million years old, first things that move. So here's a Dickinsonia specimen, and here's a footprint right next to it. So that it's positive and negative, and you can even see the modules in a lot of these. So we have beds where we have like 13 footprints and end with a Dickinsonia as it moved across the seafloor. So we have the advent of mobility. The reason we can tell this guy moved was because of its interaction with the mat. By absorbing and removing the mat and leaving a divot on the seafloor that the sand then fills, gives us evidence that it moved. This is just another guy right here or here in the stamp series that was like a snail today and would scratch at the mat. Here's another thing that looks a little bit more familiar, right? This is an organism that moved on the seafloor and grazed and ate the mat. This is something we know was made by our closest relatives because it's something that moved through the sediment, moved by peristalsis, like many worms do today with a fluid skeleton when you see an earthworm. For a variety of reasons, we know that it was a bilaterian. We are bilaterians. We are bilaterally symmetrical. We have a through gut, a head, an anus, dorsal and ventral side. So there's another bilaterian. We knew about this trace fossil for 2 decades. We always were like, oh, I wish we'd find what made it. Excavating like bed 35, we find a whole pile of these little things that they look like smushed, if you took a rice grain and smushed it in mud, only a lot smaller. So we then got a laser scanner, as you see here, 2 very happy looking chaps. The one on the left is my kid. Anybody who does fieldwork knows that when you have highly technical things like laser scanners and you're working with them in the field, 2/3 of the time, as my student Heather knows, who's here, they never work. And so it's always just, and these are like $50,000 pieces of equipment. So very scary. But hurrah, we found this guy here, which we named Acaria, which is at Namutna for meeting place, the oldest bilaterian. It had modules. Here is a great reconstruction by one of our graphics people at UCR. And you can see here's the fossil, so on, head, end. So oldest bilaterian, moved, very big deal. So one of the things that's cool, again, going back to Dickinsonia, I was talking about the mat, is the relationship of the fossil with the mat. So we know Dickinsonia moved. Look how sharp that is, right? That's really sharp. But not everybody has sharp edges. So this is a fossil we named a few years ago called Obamas. If you look at the edge, it is not sharp, right? You can barely make out, this is the fossil, this is a silly putty showing you what the animal looked like. It sort of is very smooth going into that, and you're like, maybe the edge is there, but it's sitting in the mat, and the mat's growing around the edges. Same with this guy here, Rugoconides, a little bit sharper, but still not that crisp Dickinsonia. Why is that important? These guys, we can say, yeah, they weren't mobile, they were just sitting on the seafloor. It's important when we look at things like this. So that's another post-it. This is something that we nicknamed raisin. And I had a couple of raisins on beds, but only a couple, so I ignored it. Because I'm like, who knows? Then we excavated a bed that was covered with these, and then we had to, of course, deal with it. What you notice looking at it is very sharp edges. Here, one of my students, Heather McCandless, who's here tonight, looks to have all these ridges and so on, but there's no regular pattern to these ridges at all. What Heather did and what we've seen using Blender is that they really do, if you sort of take the raisin grape analogy, that these are deflated organisms that were in the water column and looked something like this, sort of ctenophore grade. We get to this point by backing out, how do you get this raisiny thing in the sediment and what can we interpret from it? And the way that we can do that is because we have hundreds of specimens sitting on a seafloor. So just to show you a couple of other new fossils, you can see a question mark here. This is a new fossil we called Quaestio simpsonorum. This is from this bed here. This particular diagram, this was in Apple News Conversation Starter a couple months ago, so on your iPhone. What is the purple blob? So this is another mobile organism. It is otherwise bilaterally symmetrical, but then has this question mark on it. So one of the first organisms to exhibit chirality, or taking something that's bilaterally symmetrical and a new feature that is not. We have that as well. Most animals do. Then this is the same bed, again, the beautiful scenery of South Australia. where we had this specimen we found here. These are scans. We nicknamed it fishhook for obvious reasons. We had one fishhook, and we were like, I wonder what that is, huh? Then we found 100+ fishhooks, all looking a little bit like this. This is a laser scan of this guy. With using a variety of tools, most importantly, is everything here, as I've said, is soft-bodied. This guy is also soft-bodied, but you can imagine like a worm gets buried by sand, it goes, and flattens. This guy isn't flat. It retains its whole circular diameter. It does this sort of sinuous turn, also has trace fossils, little burrows where it moved, and we interpret it as an ecdysozoan. Ecdysozoa is the group of animals that includes things like insects, crabs, and nematodes. And working with our fabulous nematology department at UCR, and they were all very excited and like, yes, 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 it's a nematode. So possible nematode. And might well be a nematode. This was a fabulous reconstruction that literally magically appeared on the Wikipedia page. We had nothing to do with the Wikipedia page or this, but it captures it beautifully as we would have done it, only not as well. So, whoops, I skipped. There we go. So I've talked about a lot of individual fossils. What did the ecosystem look like? This is a reconstruction by former student Walker Weiland. What did the seafloor look like? What do these communities look like? So just to give you a couple of examples, this is work done by my student Phil Bowen, who's here. I don't know about your yard, but my yard has tons of little palm fronds that are about this high. And you spend all your time pulling them out, or I do, or I don't right now. But you know that at some point, only one big palm tree is going to come out of that, right? It's something called self-thinning. It's really common in forests where you have tons of little sprouts, but only one is going to grow up to be the oak tree, right? Very common in coral reefs, not known otherwise from the fossil record until Phil did this study here, because remember we have seafloors, so you can look at all aspects of the way that organisms are interacting. And here, this frond guy, which is sort of like a sea pen, and what Phil has recently been able to show is that you get lots and lots of these little guys, and that they self-thin, and only a few bigger organisms coming up. The reason we can do work like this is because unlike the rest of the fossil record, we're actually sort of looking almost at a modern ecological scene. And then one more, because this is like a very happy way to end, is you see purple dots and yellow dots. The purple dots are Dickinsonia, which you now know very well. The yellow dots are a thing called Sparginomorph. It's a tiny little like pill bud kind of thing, the little roly-polies. And this is also work by Phil, and what Phil has been able to show, and these are, this is about a 20 square meter bed, is that these little guys all line up behind the Dickinsonia, and I don't know if you can see the little guys here. Here's one, here's one, here's one. And they all line up just behind and on the side of the Dickinsonia, facing the same direction. And so we've interpreted this as commensalism, which is like a really nice thing, right? It is the Dickinsonia is feeding as it gets up. You can imagine all that organic stuff gets up in the water column and gets loosened up, and that these guys are eating it, which is pretty amazing that we're able to even think about things like that in rocks that are, in fact, 550 million years old. There is no predation in the Ediacaran. Nothing had teeth. We have no evidence of predation. It's often called the Garden of Ediacara for that reason. And so this sort of fits into that lifestyle. So what can we say about Earth's earliest ecosystem? We have the dawn and early evolution of animal life, and we have to go at it in a really sideways sort of way to get at what these organisms were doing. And in some, or many of them, we just know how they ate, we know how they moved, but how were they related to animals? We don't know. In others, we can really start to put them on the animal tree of life. Ecosystem is way more complicated than I would have imagined, and that I think anybody would have imagined. And that we start to see interactions between and within species. And I think the take home here is that when the conditions were right, complex life really took off. We had about 4 billion years of Earth's history without complex life. And in this case, we think there was an increase in oxygen. And when that happened, life found a way, to quote Jeff Goldblum. So, as Peter mentioned, this is now a new national park, Nilpena Ediacara National Park, with a great AV system and all of that. So if any of you guys find yourself in South Australia, come visit. We are the 2026 Australia World Heritage bid, which is very exciting and a little nerve-wracking. So we'll see how that goes, but we're hoping. South Australia only has half a World Heritage site with a vertebrate fossil locality, literally only a half. The other one is in Queensland. So they're hoping for a whole World Heritage site with this. So obviously this kind of work takes a huge number of people. I used to have a slide that had names listed, but there were so many names, it was just a blur. So now I just put a few pictures. And it shows you some of the beautiful scenery that we have there. So I'll end with this. This is a photograph we took at Nilpena Station. That is one of the old historic buildings there, looking out to the sky and looking out to the stars and thinking, you know, is life evolving on some other planet other than this one? And if so, I don't think we're going to find a Dickinsonia, but what we do learn from here is how life evolves. What are the physical and chemical processes and things like that, and like microbial mats and whatnot. So I will leave it there, and I think my next slide asks for, yes, questions.
So now you would not be surprised to learn that Mary is the recipient of 3 prestigious medals. In 2022 from the National Academy Walcott Medal, in 2022 the Moore Medal, and then in 2024 the Paleontological Society Medal. And that is the fact that she's been recognised nationally as a preeminent scholar in a field after a presentation like that, and seeing the impact of her work. It's not surprising, and congratulations. So, as she said, she will now take questions. If you have a question, please raise your hand, and one of our ambassadors will present you with a microphone.
Maybe it's just my inability to look at some of your diagrams, but it looked like they were all devoid of plant life in there.
Yes. You mean the outback, or?
In the outback, right.
Well, it's like here. There are some, you can see in this picture, this was taken there. There is saltbush, which is sort of...
No.
Oh, the fossils.
The fossil plant
Okay,
life.
sorry, that's why I was like the outback, or the...
Yeah.
Yeah, okay. Yes. Okay, so we have, it's underwater, so it's the ocean. Agreed. So we wouldn't, there's no life on land yet, maybe some like scum kind of stuff, but no life on land. It's the ocean floor. We do have algae. So we have, in terms of things that photosynthesize, we have the organic mat, and we do also have algae, which in fact Heather is working on. But no sort of plants that you would be familiar with, because it's just like being on the modern seafloor.
Agreed, but you'd think that you'd see fossil remnants of the plant because we find fossil remnants from 3.5 million years ago.
Yeah, so, but
So-
it's before there's life on land. So, but we do have fossil remnants of algae, which of course give rise to plants.
Yeah, stromatolites and
Yeah, so, yes, so stromatolites are microbial. So the microbial mats that I was showing, the organic mats,
right.
that's the equivalent of that in this kind of rock. So this,
and you see those in the rock.
oh yeah, that's what I was saying.
Okay,
That,
that's what I
that
say.
is your
Looking-
organic mat,
Looking at
yes.
the pictures, you couldn't see any.
Oh,
anything
that,
like
when I showed you the green scum and then said, this is what the rocks
I
look like.
agree about the renderings, but not the rocks.
Yes, so this, I'll just go back a second. Well, even this, right? So this here, this sort of textured surface, that is the reflection of organic mats. So we have stromatolites in rocks above and below, but they're carbonate. These are sandstones, and you don't get stromatolites in sandstones.
Yeah.
So this is what we get instead.
Okay.
Yeah, sorry I didn't understand the question to begin with.
I'm back here.
Thank you.
I love those postage stamps. It makes me think makes me think that snail mail has a new definition in origin. Can you talk about the maybe even premature origin of the Ediacara in terms of evolution? Because it must have evolved from something.
Yeah.
And over maybe 550 million years, what it may have evolved to in terms of, say, present
Right.
organisms?
So, both sides of this. So there are, as I said, Ediacaran fossils older than this by, say, 5 million years or so. They are all much of a muchness, as I would say. They are built, they are sort of fractal. They're built like our lungs are built, so maximizing surface area. And so the things that I showed you that look sort of like sea pens, right? So that's the oldest record. Then we think there was an increase in oxygen and we get this record. The overwhelming majority of these guys go extinct soon after this, but a handful of them go through, including, we would say, Acaria, the little tiny thing that made the burrow that I said was a bilaterian, the ecdysozoan, the fish hook. So a handful of these guys make it through that mass extinction and would have relatives living today. The problem is, of course, it isn't obvious for the most part. And most of these certainly, we have a number of things that are triradial. They absolutely went extinct. There is a mass extinction that happens a little after this, but just enough made it through to give rise to what we have today. Does that answer your question?
Outstanding. Many years ago, I had an opportunity to visit the Burgess Shale, and I don't know how the continents aligned back then, but how does what you discovered map against the Burgess Shale?
Yeah, great question. So the Burgess Shale, and lucky you, the Burgess Shale is younger than this. So this is, the Burgess Shale is part of what we call the Cambrian explosion. Or Cambrian radiation, but Cambrian explosion, cover of Time magazine, Cambrian explosion, where you get a huge diversification of animals and lots of like arthropods, things like trilobites, things with skeletons, and a lot of soft-bodied organisms that are preserved in the Burgess Shale. That is like 20 million years younger than this. So the way that we think about it is that the Ediacaran is the time of experimentation, evolutionary experimentation with, say, body plans, right? Here's a triradial, here's a thing with modules, and so on. Many of these went extinct, some made it through, and then you have the Cambrian explosion, which is like huge diversification within, say, arthropods things related to crabs, clams, crabs, lobsters, insects, and so on. So the Burgess Shale is that next step. We have lots of arguments with Cambrian people because they think theirs is more important. We're like, no, we're the beginning, you know. So yes, good question. Thank you.
So I have one question, and it's not scientific, but from your efforts, there's a national park there now, and
Yep.
now obviously it's Australia's nominee for a World Heritage Site. You just want to reflect on how that came to be, you know, and the work involved. It's probably impossible to summarise in 1 or 2 minutes, but you know, there's a national park because of what you've been working on, and now you know, international establishment of a World Heritage Site. It's astonishing. It's not surprising in terms of the implications of what you've done, but then these types of things I think are astonishing.
Yeah, so incredible journey. I mean, 5 years in, Jim, my colleague who's since retired at the South Australian Museum, we knew we had something special. Ross Farger, who I showed a picture of, who is the landowner, really leaned in heavily and protect and conservation. One of those, a miserable specimen of Dickinsonia goes on black market, eBay, $5,000. We have thousands of them. So this is also a big security issue. We started talking to groups around 2010, you know, trying to see who might be interested in something here. And then the then Premier of South Australia, a guy named Jay Weatherall, brought his country cabinet up. So, you know, it's the Premier, so like a governor brings all his ministers or her ministers up to sort of outback regions. And so he was coming up to this outback region. I literally flew for the weekend back to take him around because Ross and his wife Jane were like, can you just come and talk? So I went for the weekend. I also told him that at the time my daughter, who was in 8th grade, had learned about the Ediacara in Gage Middle School in Riverside. You know, biology teacher talking about the Ediacara, and she comes home, she's like, you know, it's like a cool mom moment. My kids have been dragged there every summer of their life, and she's like, hey, Mom, we talked about... So I told that story, and Jay Weatherall walked away and was like, wow, we're going to do something here. And that started the process. The amazing thing from me was that was a Labor government. A Liberal government came in soon after that, so sort of the Conservatives. This park and everything about it went seamlessly through those and back out the other end with the Labor government who opened this. I became very good friends with the Premier, the first 2 premiers who would come up, and they just got it and realized this was something very special about South Australia. Amazing. With the park, I would come home from work. I was living on Australia time. I would start doing Zooms at 5 o'clock and finish at 10 p.m. We got special permission, went through millions of hurdles, scary police escort, all sorts of things to go during COVID when Australia was otherwise shut down so that we could develop this park. You know, I have such good friends there, and it has been a very long, arduous, but really fun experience, but mostly working with the government there and traditional landowners, the Adnyamathanha, we do a lot with them, and pastoralists and tour operators, and everybody sort of coming to the table with a common cause. It's hard to imagine that can happen, but it did. So yes, answer your question.
Any more questions for Professor Droser? Yeah.
Hi. In other parts of the world, are you finding similar specimens?
Great question. So there's only one place in the world where you find this suite, and that is the White Sea in Russia, which is these sort of clay, fine-grained beds right on the cliffs of the White Sea that sort of fall in, so they can't do anything like this. In Newfoundland, there are fossils that are older, and in Namibia, there are fossils that are younger. Those are the four sort of, and China, quite really good localities. There's scrappy fossils all over out in Nevada. You can find some really scrappy fossils. But this is the only place this is the only place in the world where we've had any excavation of sort of fossil beds of any age. So it's absolutely unique. So it's not that this is going on, it's that these fossils are found elsewhere and clink, 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 taken back to the museum and so on.
Any more questions?
Thank you. It's really interesting, and I'm totally a novice. But about 30 years ago, when I got to go with people who are excavating blue stone that is used to build Wall Street, which is now kind of, they don't mine it anymore, but we saw a lot of things that look like that, but I had no idea like how old they might be or if they're of any consequence, scientifically, because it's upstate New York, and sure there's been no water there for God knows how long.
Yes. Yeah, so they would be much younger than this. You can do like sort of fossil and geology tours of buildings in New York City, but they would be they'd be, say, 50 to 100 million years younger than this. So with not soft-bodied things like this, but they do have fossils in them. Yeah.
Okay, if there are no more questions, let's please thank Professor Droser again for a fantastic presentation. So that concludes this evening's Science Lecture Series. Thanks again for coming out. Please join us for the second instalment of this series, which will be here at the Palm Desert Centre on Tuesday, March the 4th, with UCR Riverside CNAS Assistant Professor of Physics, Miguel Arratia, who specialises in experimental particle and nuclear physics. His presentation will be on the quantum tomography of the nucleus with the electron-ion collide. So to translate that into everyday speak, if you have ever wondered what a nucleus of an atom looks like, and I suspect it's not little spheres with P and N written on them, right? What it really looks like, then Miguel will take us through his own journey to actually show you what the state of the art is in the visualization of basic atomic matter. So thanks again for coming out, and look forward to seeing you in a month's time.